Hi, my name is Joe Fox. I'm a professor at the University of Delaware working in the Department of Chemistry and Biochemistry. Work in my group is broadly focused on developing new types of chemical reactions for applications in biology. And along with my students, I'm going to talk to you about our work to develop fast and inducible methods for bioorthogonal chemistry. Okay, so bioorthogonal chemistry represents a class of non-natural reactions that can take place selectively in biological context. This is in direct contrast to conventional substitution reactions, which are not selective enough to occur in a living system. This panel outlines 8 major classes of bioorthogonal reactions. A number of years ago, our group was the first to describe tetrazene ligation, shown here in the yellow panel. And a major feature of tetrazene ligation is its ultra-fast kinetics. This panel outlines the mechanism of tetrazene ligation with its most reactive dienophile transcyclooctene. Transcyclooctene is a strained alkene, and our group has used computation to design a number of variants in order to improve the properties of transcyclooctene, including its reaction rate. Notice here on the right, we've designed analogs with reaction rates as fast as 10 to the 7th per mole per second. That translates into some really exceptional kinetics with complete reactions with quantitative yield within 12 milliseconds using only 160 micromolar in the excess reagent of tetrazene in this case.
Hi, my name is Jessica, and I'm excited to share my work involving the development of a new TCO synthesis. Since 2008, the Fox Lab has synthesized TCOs by a photochemical flow method, irradiating cis-cyclooctenes and receiving a mixture of diastereomers. These diastereomers have different kinetic properties, can be difficult to separate chromatographically, and the minor product is often the more desired. I've developed a new synthetic platform using a ketone TCO precursor that, when reacted with nucleophiles, forms a single diastereomer. We have found that these new TCOs have improved kinetics and physiochemical properties, and that we can form a library of compounds from a single ketone starting material.
Hi, my name is Bill Lambert, and I'm a recent graduate of the Fox Lab, now working at AbbVie Pharmaceuticals in Chicago. So in Joe's group, I helped develop a safer and broader tetrazene synthesis through a cross-coupling approach, whereas traditional tetrazene synthesis was often hindered by hazardous reagents and limited functional group tolerance. And so here we designed a reactive tetrazene reagent, which we called BTZ, for the first ever silver-mediated Libeskind-Sorogo cross-coupling reaction with boronic acids. The reaction is broad, tolerating a large variety of boronic acid functional groups, including in the synthesis of tetrazene fluorophores and tetrazene drug conjugates. This reaction has also been made widely accessible by commercializing BTZ through our partners at Sigma Aldrich. And we hope that with this approach, in conjunction with other approaches and methodologies developed in the Fox Lab, that these tools are more widely used in the field of chemical biology.
My name is Yixing. I would like to introduce my work to develop tetrazene-based biocycler tools. I developed a one-pot synthesis of thiomethyltetrazene from carboxylic acid precursors involving Lewis acid catalyzed rearrangement, condensation with methyl thiohydrolate, and oxidation to target tetrazene. Thiomethyltetrazene provides a divergent platform for functional tetrathenes. Monosubstituted tetrazene can be synthesized through palladium catalyzed reduction using Libeskind coupling with boronic acids or standing reagent. Unsymmetric tetrazene with aliphatic, aromatic, and heterocyclic substituents can be propelled. Advantage of these approaches are without direct handling of hydrogen and broad functional group tolerance. Also, small and hydrophilic tetrazene can be conjugated to bioactive molecules. A series of tetrazene Dept probes and tetrazene mega inhibitors are now investigated by our collaborator at Pfizer.
This panel describes a practical new method for integrating protein science with bioorthogonal chemistry. It was developed by my student Sam Sinto, who you'll be hearing from shortly. One of the great advantages of tetrazene ligation is that it allows you to attach essentially any transcyclooctene to a protein of interest bearing a tetrazene handle. The issue has been that site-selective methods for tagging small molecules such as tetrazenes have really been lacking. Sam found a way to address this issue by developing the concept of ABC tags, affinity bioorthogonal chemistry tags. Here, the tetrazine serves not only as a bioorthogonal group, but also as a purification handle, because it can coordinate to the nickel resins that are commonly used for protein purification. You can see that it works extremely well to clean up even very messy reaction mixtures, as shown on the right, and then can subsequently be used in quantitative tagging with any transcyclooctene of interest. Our group has also used tetrazine ligation as the basis for the construction of a range of probes for biological applications. Shown in this panel on the left is our work, some of our work to use tetrazine ligation to construct probes for F-18 positron emission tomography imaging in live animals. This has been a collaboration with the group of Zibo Li and has been effectively used to construct probes that can be used to image tumors in live animals. We've also used tetrazine ligation to construct probes for imaging reactive intermediates called sulfenic acids, which my student Sam will tell you about, as well as for developing diazarines for photoaffinity labeling, which my student Jess will tell you more about.
Hi, my name is Sam. I have two projects that I focus on in this lab. One is using transcyclooctenes as sulfenic acid probes. So this would be a small molecule probe for oxidized cysteine, with the end goal being for chemical proteomics.
Hi, I'm Jess, and I'm a fifth year student in the Fox group. My current project is the development of a true carbene dialkyl diazarine. The goal is to have a diazarine that can do CH insertion. And so my project is the interface of chemical biology, organic synthesis, and also photochemistry.
Another exciting new direction for my research group has been the development of new approaches to activating rapid bioorthogonal chemistry. Here we use catalysis, either photocatalysis or enzymes to turn on the tetrazine ligation. With light, we call this the catalytic activation of bioorthogonal chemistry with light or cable. And we start with the dihydrotetrazine shown here, and we've developed a number of really exciting photocatalysts based on common fluorophores like silyrotamines and even fluorescein, which in response to long wavelength light can catalyze the oxidation of dihydrotetrazines to tetrazines using oxygen as a terminal oxidant and producing peroxide as the product of the reaction. It's really beautiful. The starting materials are yellow and the products are bright red or pink. And it's also very useful. So in one manifestation of this, we've learned that we can use this to cross-link materials, and we can actually do this reaction, this photochemical reaction, in vivo, because we use photocatalysts and wavelengths of light that are tissue penetrant. So here we take a material, a polymeric material that is a liquid, and we derivatize it by both transcyclooctene and dihydrotetrazine. And upon photocatalysis under very low concentrations of catalyst, we see that we oxidize the dihydrotetrazine to a tetrazine, which can then undergo Diels-Alder reaction to cross-link the material, which in this Violex inversion experiment, you see you initially start with a liquid that becomes a freestanding hydrogel afterwards. If you start with, you can suspend cells in this liquid and they become trapped in the hydrogel where they can be cultured. So this is a method for cellular delivery that we have been learning how to apply in living systems. So here is an experiment in live mice where we subcutaneously inject this mixture as a free-flowing liquid, and then we can create a material in vivo. You can see what this looks like after it's removed by image-guided surgery. And we envision this as a new therapeutic tool for introducing either cells or drugs for slow delivery to diseased tissue and joints.
Hi, my name is AJ, and I'm here to talk a little bit about my work in Joe Fox's group. The reaction between tetrazine and TCO is currently the fastest known bioorthogonal reaction, with its speed being driven by the electron deficiency of the tetrazine ring. However, while the most reactive tetrazines possess very high rate constants, they also exhibit very low stability in the environment of the cell. In order to circumvent this limitation, our lab developed a reduced form of tetrazine, known as dihydrotetrazine, or DHTZ, which is unreactive towards TCO. DHTZ is able to have its full reactivity restored after being reoxidized to tetrazine through the use of photocatalysis. In the presence of photocatalyst in blue-green or far-red light, DHTZ is able to be rapidly converted to tetrazine. This allows for the on-demand spatiotemporal generation of the most reactive bioorthogonal compounds. My research has focused mostly on using DHTZ in the context of the cell and for protein conjugation. My research has shown that DHTZ is very stable in vivo, as the reducing environment of the cell is able to maintain its unreactive form. I've also shown that this compound is able to be fully reoxidized using photocatalysis in as little as 1 minute. The high reactivity of the nascent tetrazine means that very low concentrations of fluorescent reporters can be used for analysis of tagged proteins, in some cases as low as 50 nanomolar. As you can see here, I can utilize this technique to label proteins in the actin, mitochondria, and nuclei and cytosol of live cells. I've also used 2-photon microscopy to spatially activate proteins in a subcellular level to write letters onto individual organelles.
Hi, I'm Jules, and I'm excited to talk to you about my project involving drug delivery via subcellular photocatalysis. In my research, I've been able to localize a photocatalyst to the nucleus, and by using our prodrug system outlined here, I've been able to target drug release selectively via the localized photocatalyst and shining red light. So you can see only when we have light and photocatalyst present do we get drug release, and the specific drug we're releasing depolymerizes tubulin, so you can see the shrunken cells only with light present, but without light, we do not get drug release, so you can see the cells are pretty happy and healthy there.
Hi, my name is Amanda Talon, and I'm excited to introduce my project to you. So my project is centered around the enzymatic activation of the tetrazine TCO ligation by genetically encodable catalysts. Our group has determined that horseradish peroxidase can oxidize dihydrotetrazine to tetrazine. However, one of the limitations of utilizing horseradish peroxidase as a genetically encoded catalyst is that horseradish peroxidase is inactive when expressed in mammalian cytosol. Apex-2 is an engineered ascorbic peroxidase that can also oxidize dihydrotetrazine and is active when expressed in mammalian cytosol. So my project's focus is to utilize Apex-2 as a genetically encoded catalyst to oxidize dihydrotetrazine and turn on the tetrazine TCO ligation. I've done some site-directed mutagenesis to improve Apex-2's activity with dihydrotetrazine, and now I'm currently looking at Apex-2's ability to oxidize dihydrotetrazines intracellularly.
Thank you so much for taking this opportunity to learn more about our research group. If you have any questions, please don't hesitate to email me or any of my students. Thanks again.